so we'll talk about the role of mechanical morphogenesis on the development and evolution of neocortical organization. And the main question that I would like to address is how do we get to develop a cognition? How do we become aware of a world of what surrounds us? No, and the philosopher Heraclitus in a long time ago proposed that maybe the logos, so the ability of understanding things, would be like a thunder, no? A thunder that, for example, in the night would shine a light on a place, and then all of a sudden everything will become part of the same perception. I prefer this image, which is the light entering through a window, and all of a sudden all the objects that get this light get to be bound together into a single coherent perception of a home, of a certain time, and a certain nostalgia potentially. So it's very difficult to try to understand cognition because we are always living within cognition, so we cannot take a step back from cognition. I think, however, what's interesting is that we can try to understand brain development, so the development of this cognition. So instead of trying to address directly the problem of ontology, try to address the problem of ontogenesis. So we may not be able to take a step back from our own thinking, but we can look at the process where a single cell can turn into an organism that's able to develop a world, a creativity, and awareness. So I'm particularly interested in the shape of the neocortex and the relationship, the development of this shape. And this shape is very interesting in particular because it shares a relationship with the organization of the neocortex. This is the map that Brodmann produced at the beginning of the... At the beginning of the 20th century, and you see that he was already noticing a relationship between the shape of different brain folds in the neocortex and the cytoarchitectonic, so the histological organization of the different regions in the brain. So, for example, one part of the neocortex will have a different number of layers with different thicknesses, with different types of cells. And very often, as it's the case of the central sulcus, I don't know if you're able to see the mouse of my computer.
So, Professor, maybe you can turn on the pointer in color. In the you can do click in the side and right side of your mouse. Okay, I will. I'm not using PowerPoint. I'm using Keynote, but I seem to remember the place where that could be changed. Show. We'll take just a second. Show the pointer with links or movies. Show the pointer. You let me know. Let me know if now it works. Okay. I will share my screen again. Now even I can't see it. Okay, so that wasn't the right approach.
Go to Keynote Preferences from the taskbar top of your Mac. Use the hotkeys to open Preferences manually. Click Slideshow tab. At the top of the Preferences window, select Show Pointer when using mouse or trackpad.
Okay, that's what I have.
Okay.
Okay, I think it will not be larger than this, unfortunately.
Maybe you can try in there, the right. You can click the right in that moment, in the right of your mouse.
I'm using a trackpad.
Right.
Ah, oh, sorry. Okay.
Okay.
Or control click.
I will try control click. Oops. You get to see the beautiful image there.
No, nothing.
No, it's not working. It's not working. Well, I will try just to describe with the verbally. So there's a very deep fold in the human neocortex. That's the central sulcus that you can also find in several other primates. And this fold that's very deep and develops very early, it also separates two different cytoarchitectonic regions in the neocortex, the motor cortex from the somatosensory cortex. And it's not an isolated case. There are many cases of deep folds which also have some sort of cytoarchitectonic correspondence, not only for the cytoarchitectonic regions, but also connectivity. So it's not infrequent that you will observe what anatomists call U fibers that will connect one gyrus with the one that close by. So there's this very puzzling relationship between brain folding and brain organization. Here, there's another example. This is figure A, is the brain of a raccoon. What you see with the solid lines is the somatosensory cortex of the raccoon, the region that's associated to the forepaw of this animal. What you see in figure B is the different parts of the forepaw of the raccoon. And if you stimulate those different regions, you elicit neural activity in the corresponding regions in the neocortex. And there you see them labeled with the same numbers. Now, what's interesting is that you see that there's folds, for example, that will separate the finger from the palm or one finger from the next. And this is the case for this raccoon, but it's also the case for several other raccoons. So even though the folding pattern is not exactly the same all the time, the correspondence between brain folding and the somatosensory fields associated to different parts of the forepaw of the raccoon is conserved. So there's also a kind of a functional relationship between functional organization of the neocortex and anatomy. It's also interesting that if you observe the neocortex of mammals of different sizes, so here we are going from the left, the neocortex of the rat, and this is showing the different visual areas, to the neocortex of the cat, to the neocortex of the macaque, and you see that in addition to a degree of folding that increases as you move from rats to macaques, also the complexity and the number of cytoarchitectonic areas associated with visual function also increases. This is shown in a more synthetic way in this plot, where you see in the x-axis the size of the volume, brain volume in logarithmic scale, and in the y-axis you observe the logarithm of the number of cytoarchitectonic areas. And there's a quite interesting relationship. So brains, as they grow larger, they grow more complex in terms of cytoarchitectonic differentiation, and they also turn more and more folded. The normal, almost classical way already of explaining how you can create new cytoarchitectonic regions is illustrated in this review by Dennis O'Leary. So the idea would be, and this comes mostly from experiments done in mice, that you have molecular gradients, different genes which are expressed in gradients over the neocortex, and that's the image that you have on the left. And then those different molecular gradients would provide some sort of coordinate system. And then a genetic program would read those coordinate systems and produce the different cytoarchitectonic regions that you can observe in the little schema to the right of your screen. The problem is that we at the moment, we haven't found evidence, as far as I know, of what would be these mechanisms that are able to transform a gradient into discrete cytoarchitectonic fields. So this, to me, it's reminds me of a very prescriptive model for how a brain would be organized, which is not unlike what used to be the explanation, the normal explanation for the way in which planets know what's the orbit they should follow in space. So here you have the classical medieval idea illustrated in this manuscript from the 14th century, where angels would prescribe the direction that planets should take. this paradigm was in part replaced by the Newtonian idea of forces, which was able to explain the orbits as some sort of emergent property of different planets and moving influenced by gravity. So we don't need anymore this kind of angels prescribing or guiding what the evolution of the planets in their orbits would be. So I would like to talk about this concept that tries to be inspired by this type of idea for the development and evolution of the neocortex. And it's the idea of mechanical morphogenesis that we have been trying to develop for some time already. So the mechanical morphogenesis is this process in which physical matter, non-biological matter even, can produce very complex patterns with intricate shape. Like this is the case of snowflakes, as you can see here in this image, and without requiring actually any genes or any sort of prescription of that sort. This is only the result of mechanical instabilities produced by the structure of the water molecule and then the changes in temperature that these molecules undergo when they fall from the higher parts of the atmosphere. These are kind of my snowflakes. So here again you see the neocortices of different species. You have from the top left to the bottom right. I will not name them all, but there's a ferret, a lion, a bear, a rhinoceros, and then several different primates on the bottom row. So how does this shape come up to be? And then again, the relationship between this shape and the organization of the neocortex and the cerebrum. So the approach that I will take to try to explain this is inspired by elasticity theory and then the experiments that physicists have been doing on inanimated matter to try to understand the process of mechanical morphogenesis and the emergence of shape out of mechanical forces. What you observe here is just gels. So you have one blue gel on top of elastic that's on top of a light blue gel. The dark blue gel is growing because it absorbs a solvent that you add to the system, and then to release the intrinsic stresses that the growth creates on the system, there's these folds which appear, which are very reminiscent of the folds that you can observe in gyrocephalic mammalian brains. The idea that mechanics and the process of mechanical morphogenesis can have an influence on the development of not only the brain, but the development of organismal form is not new. This is, for example, the work of Wilhelm His in 1894. And he's very well known as being the inventor of the microtome that all neuroscientists would know very well. But he's also well known for his work on the developmental mechanics theory. So he was trying to come up with mechanical systems that would exhibit some of the properties of a developing embryo. Here, for example, he was comparing the formation of different somites in the embryo with the convolutions in a piece of leather that would be nailed to a curved piece of wood. And so briefly to introduce you to the way in which deformation is conceived in elasticity theory, let me tell you just in a very intuitive way, how do we deal with elasticity and with growth in a biomechanical model. So the main idea is that you will decompose deformations of a tissue into two basic types of deformations. So to the left of your screen, we have changes in shape, which do not alter the volume. So the volume of the piece of tissue on top is the same as the piece of tissue at the bottom. The only difference is this skew that will just change the shape, but not the volume. On the contrary, you can have also a change in size, so a change in volume without a change in shape, which is the example to the right. The idea is to represent this type of deformations as a kind of energy. So in different materials, there will be an elastic energy that will be differently affected by changes in shape and by changes in size. So you represent this W, the elastic energy, as the sum of how much shape changes plus how much size changes. And the type, depending on the type of tissue, the values of mu and kappa will be different. So for example, in the case of biological tissue, like brain tissue, it's a largely incompressible type of tissue, so it's very difficult to change its volume, but it's more easy to change its shape. So kappa here will be a very large value and mu will be a smaller value. In mathematical terms, you represent this as a function of the deformation tensor for each of the elements of your material. Here you have, for example, the decomposition of a rectangle of elastic tissue into little elements. Now, if we want to represent the deformation of this rectangle as the strange shape that I drew at the bottom, you see that the idea is that you represent each of the elements, then you look at each of the elements, how to best approximate the type of deformation, and you have in the right of your screen something that would be a pure change in volume, a pure change in shape, and a combination of both. That's C, D, and E. And you can read more about that in our paper with Ophélie Foubé. And now how you add growth. This is only elasticity, but now we have growth. If you remember from high school, when we were talking about elasticity, you have, for example, imagine a piece of a spring, an elastic spring. So if you don't touch your spring, it will have a rest length. No, it's the size of the spring when no one is touching it. After that, you can pull or you can push your string to make it larger or to make it shrink. And as soon as you release the spring, it will go back to its rest size. So the idea for the modeling of growth is that you imagine that you have some sort of a force that will change the resting size of your elements of volume. Okay, so here the size at rest is represented by this wireframe, and now you see that because of the forces of growth, the resting size of this element of volume, it's larger, which will make the piece of tissue grow. Okay, and you combine both. You combine F, which is the deformation tensor that we obtained before from elasticity, with the growth tensor, which is G, that it's the one that we obtained from growth to obtain a total deformation, which is the tensor FT. And this is the seminal work of Rodriguez and collaborators in 1994. That's more or less the basis of all the modeling of growth that physicists do right now. After that, we need a way of implementing this growth model in a computer. And very recently, there's been a lot of development in ways of modeling different materials, and this is the method that we are using. It's called the material point method. That's very interesting. So there's always this kind of balance that you need to have between, do I model my system as a combination of particles, or do I model it as some sort of a grid? So very often you will use a grid to model flow of or a gas flowing. For example, if you wanted to model a river, it would make more sense to try to tell how much water is passing a square centimeter of the river than trying to simulate each of the water molecules in the river. So that's what's called the Eulerian approach. And then the approach that's more used, for example, if you want to model planets, you will represent each planet by a particle. That's known as the Lagrangian approach. What's interesting with this material point method is that you are permanently going from one approach to the other. So here in figure 1, what's easy to model as particles, you model as particles, but then you move all your particles into a grid. You do everything that's easy to do, that would be 2, 3, and 4, you do in the grid, and then you go back to the particles, and then you repeat again and again and again. And in this way, you can have very convincing simulations, pretty accurate, of different types of materials. Could be liquids like water, can be gases, but can also be soft elastic tissues like biological tissue. Let me show you just one simple example where you will be able to see the little particles. This is a very basic model of a system, but that would resemble a cerebrum. You have the red particles, which compose what could be the neocortex, and the green particles, blue particles, That could be the subcortical substance, be it radial glia or white matter. What I have added is elasticity and growth, and because of the action of growth, every particle is growing in the same amount. You observe this folding that appears. And now this is the type of folding that we can obtain if we use the same type of biomechanical model, but in 3 dimensions, starting from something that looks like an ellipsoid. So you have a first stage where the model grows without folding. At some point, you reach the critical point where folding is more energetically economic than growing without folding, and that's where this buckling instability kicks in, and you observe the development of folds which resemble very much those that we are able to observe in brains, but also in some other type of materials. For example, there's some lemons that fold in a similar way. What's interesting is that when you look at what happens at the mechanical level in these folds, you observe, first, this is a surface map of the deformation that each region of the cortex underwent. So when you have a fold, very often on the top of the fold, there will be some expansion. So the particles or the elements in the top of a gyrus will tend to expand, but those in a sulcus will be under compression, so they will tend to shrink. Even though the growth of the cortex is homogeneous, that leads to non-homogeneous deformation of the cortex. And that's what I'm mapping here. This is the initial shape of the model to the left, and the final, the labels are swapped. Don't believe the labels, believe me. So to the left of your screen, you have the initial shape with the deformation map, and to the right of your screen, the final shape with the same deformation map. Is just that you are not able to see it because it's folded. And you can see that there's this folding pattern represented in the amount of shrinkage that each different part of the cortex underwent, despite the homogeneity of the growth. So what's interesting also is if you cut now this model, you observe this type of phenomenon. So this is from a paper by Tallinn and collaborators in 2014. And what you observe is that the formation of these folds leads to the formation of a series of mechanical gradients. Okay, so these are gradients which are not unlike molecular gradients, but here what you have changing is not the density of a molecule, but the amount of residual stress at every different part of the tissue. So very often under a gyrus there will be, which is what you see in red here, tensile stress in red is showing that under a gyrus there's a lot of stretch. So the white matter will be, it's being pulled by the growing gyri, and then what you see in the second panel. So under a sulcus, it's the figure B in the middle of your screen. This red region is a strong region of compression. So because of the formation of this fold, there was a compression of all the tissue in that region. Okay, so these are gradients which are as much as a gradient as a molecular gradient. However, they are produced at the level of the complete organ. They can be established in an organ like the brain or in any other part of the mammalian embryo at the speed of sound, which is the speed of propagation of mechanical forces. And what's very interesting, that we are more and more realizing that biological tissue, developing tissue, is very sensitive to all this type of mechanical forces. We have reviewed the literature in this paper with Ophélie Foubet, Miguel Trejo, and myself, that you can go read for more information. But here's a brief summary. So these are some of the effects that mechanics can have on developing tissue. It's well known that there's an effect of mechanics and stiffness, of mechanical stiffness, on cell proliferation. So under compression or in a rigid substrate, cells tend to not proliferate, or you can even trigger apoptosis just using mechanical properties. You can also alter cell fate. So you can, for example, make a neuronal progenitor derive into glia or neuron just by changing the mechanical properties. You can also change the cell shape. So neurons will tend to have a more rich arborization if they are being grown in a soft substrate compared with a stiffer substrate. You can also alter axonal guidance through mechanical properties. So these gradients that produced by brain folding could also potentially affect axonal guidance. Here you have a very clever experiment. What you see in panel A is, imagine spaghetti. No, it's a bed of spaghetti. Each of these little ellipsoids is like a thin, very thin road with the shape of a spaghetti. So it's very easy to bend the spaghetti in the direction where the spaghetti is like flat, but it's much more difficult to bend it in the other direction. So the stiffness of this substrate is anisotropic. It's much more easy, so it's much more rigid in the horizontal direction than in the vertical direction. And what you observe when you make axons grow in this type of substrate is that axons clearly prefer the more rigid orientation for growing. And you can also produce, trigger, and modulate synaptic activity. I will not go into the details. So we could, what I'm proposing is that mechanical morphogenesis is a mechanism that could have a causal effect on the development of a neocortex. So it wouldn't be only the pure lecture of a genetic, the pure reading of a genetic program that would define the form, but more like some sort of feedback loop that would potentially make form affect the transcription of some genes, and in turn that could affect the form. So we are trying to conceive a system where geometric and mechanical constraints influence and define neocortical organization, and also possibly the geometry of cognition. So now I will try to show you the results that we have gathered during these last years so adopting this perspective on neocortical development and later on neocortical evolution. So for neocortical development, we developed this project that was called FIND, which is the creation of the Ferret Interactive Integrated Neurodevelopmental Atlas with a series of collaborators. Our aim was to obtain data from ferrets. Ferrets are very, very interesting, a very interesting animal model for the development of the neocortex, because when they are born at P0, they have a brain that's of a similar size as a mouse brain, completely smooth, as you can see to the left of your screen. And then in the first week of development, you start to observe brain folding. So brain folding will kick in somewhere between P4 to P8. And then by the end of the first month after birth, you will have a very rich, very regular folding pattern, similar to that of a little kitten. And then for comparison, you get the brain of an adult ferret brain all to the right of your screen, and it's mostly a change in shape, but the folding pattern stays the same, and also the volume. Here you have another view, is just to let you see the folding pattern of the ferret brain. Now all brains are at the same size, and you see the dorsal views on top, and then the lateral views at the bottom. So we obtain magnetic resonance imaging for 44 ferrets at different time points. Here you see them from left to right, the P0, P2, so 2 days after birth, 4, 8, 16, 32, and then adults. We develop a series of tools for being able to produce reconstructions of the neocortex of the ferrets. This is one of our tools. It's called BrainBox, that you can access directly on the web. It's a web application for segmenting collaboratively MRI data. After that, once we obtain surface reconstructions like this one, we developed this other tool that's called Stereographic for labeling the different folds that allowed us to create some sort of reconstruction of the developmental trajectory of brain folding in ferrets. And then we developed also methods for matching those meshes so as to reduce the mechanical deformation, not mechanical, but just the deformation of moving from one brain to the other. I will not go into the details of the method. but all our methods are available online and open source. This is now the result of applying all these, and some more of these methods to the reconstruction of a developmental trajectory of a ferret brain from day 0 to the adult. So, play it again. You have this first stage where the brain grows without developing brain folding, and then this folding pattern emerges, and then folds get deeper and larger with age. So now that we have this data, we can start looking more in detail at trying to quantify the process of brain development and brain folding in ferrets. Now, what's interesting is that there's many things associated with this brain folding. We have also histological data for many of these specimens, and here you see one example of one slice of a P32 brain, and I'm showing you the neurons. I'm zooming into the neurons right under a sulcus, and then you see the effect of this compression, the formation of a sulcus pushing on that region of the brain, which leads to this very dramatic change in the shape of the neurons that you see. They are all flat. If I were to do the same in a gyrus, you would see that the shape goes in the direction predicted by the mechanical model. So neurons tend to be elongated, perpendicular to the gyri and parallel to the depth of the sulci. It's not only, of course, the neocortex. We have used methods such as those described by Pamela in the previous presentation. And here you see the connections, some of the tractography results for the ferret brains, and you see some examples of these U fibers. So these folds, we do observe connections that go from one gyrus to the gyrus next by. So it's interesting is that if we believe that what would be our main hypothesis, that actually the brain folding or this change in shape was not produced by some very precise genetic pattern that would determine where to place a fold or not, but just by a mechanical instability leading to the release of stress through folding, then it would be possible that this folding is somehow influencing the formation of the corticocortical connectivity. Okay, this is another view of this reconstruction of brain connectivity in an adult ferret, where you can see more of these examples of U fibers. Now, this is using a reconstruction of the developmental trajectories. This is a map of local surface expansion. I'm mapping everything into the same adult brain, not into the real brain. So, for example, the first figure is mapping the brain from P4, where there's no folding, to P8. But just for comparison, I'm mapping all that in the same brain. So you get to see that it seems to be more expansion in the gyri, this type of finding. So if you only do data analysis without a theoretical model behind, you could be, you may want to tell that there is specific focalized expansion of gyri. Thanks to the mathematical models that we did before, we know that it's very likely that homogeneous growth could also lead to this sort of difference between gyri expanding more than sulci, just because of mechanical instability, just because of mechanical morphogenesis. And this is the result. If we apply the same model, this is not a real brain. This is, again, a simulation of a brain. But it's the same mechanical model that I showed you before with the ellipsoid. But now, instead of using the ellipsoid as starting point, I use a shape that looks very much like that of a ferret brain. So the geometry at the beginning is that of a ferret brain. I run the biomechanical growth model, and what you observe is the formation of a very stable folding pattern that's very reminiscent of the folding pattern of a real ferret brain. The idea is that the original shape of the ferret brain somehow encodes in its geometry a potential folding pattern. So regions which are very, very intuitively. If you have regions in a brain which are closer to flat, they will be easier to fold, to bend, than regions which are more close to a dome. No, because a dome, like you may have had that experience. If you try to flatten an orange peel, it's almost impossible to flatten without breaking it. No, on the contrary, if you have a paper, even if it's very crumpled, you are able to unfold a paper because it's intrinsically flat. An orange peel is intrinsically bent. So in those regions in a mammalian brain which are closer to a flat plane, folds will be easier to produce than in those which are closer to a dome. So this is the result of a computer simulation of brain folding. And now this is the result of a computer simulation of brain connectivity. We have simulated something that looks very like a very simple brain with basically two hemispheres, or you could think it's only just two brain folds. And we have, again, a biomechanical model for the growth of axons influenced by this geometry. And you can observe that naturally, without having to enforce the presence of U fibers, they appear just because of mechanical necessity somehow. Now, of course, this folding pattern is not completely like the folding pattern of a ferret brain. But this is just a very, very simple biomechanical model where growth is homogeneous. And we know that that's not the case already. Okay, so for example, if we analyze the folding trajectory reconstructions that we had from our ferrets, this is the results that we obtained. Here we have fit in every point of the neocortical surface a Gompertz model. The Gompertz model has a shape like the one you can see to the left, bottom left of your screen. So there's an onset point, but then there will be some sort of saturation point. The Gompertz model has a parameter that can control the time of onset. So you can produce something that we call the heterochrony parameter. So you can make growth start earlier or later. And then you can also control the growth rate, so the slope of this growth curve at each point of the cortex. And that's the growth rate that you observe all to the right of your screen. So you have a gradient in the heterochrony. So different regions in the cortex will not start their growth at the same time, and they will not have the same amount of growth in a homogeneous way. And now we are working into trying to simulate this sort of gradient in growth into our folding models. The idea would be that there's a high-level control of the global growth of the neocortex, but then there's no need for very precisely specifying where each fold will be placed. That's encoded in the geometry and the mechanical constraints of the biological, the physical system that a developing brain is. This folding pattern is a very interesting point, because finally it's this folding pattern that's related. For example, there's some gyri in ferrets which are all the time the place where the auditory cortex will be located, or the visual cortex will be located, or the motor cortex will be located. There's also a very specific connectivity patterns. And for a long time, people thought that mechanical models, they would be able to produce folds, but they would be more or less random because there's no genetic program guiding them. We observed through our models that that's not necessarily the case, but we wanted to see what was the case for this through evolution. For looking into evolution, so just very briefly to introduce our work on evolution, let's imagine that this is a phylogenetic tree, for example, for primates. So you can imagine that you have all to the left our common ancestor. So the common ancestor of all primates, it's 75 million years ago, approximately. And then there's many species that may have disappeared. Those are the little branches that do not end today. And so all to the right of your screen is today. And then we have the species that we're able to study. And we do, for the sake of statistical analysis, a very dramatic simplification of this tree. And here I just want to illustrate the importance of taking this phylogenetic information into account. This will very much resonate with the presentation of of Juan Montiel not long ago. So imagine that this is, again, the common ancestor 75 million years ago. Now imagine that you have a phylogenetic tree where for a long time there were only 2 different branches, but then there was some speciation event, for example, an asteroid that fell or something like that, and then all of a sudden there's many different descendants of each of these species. Now, it's very interesting to imagine what would happen with phenotypes. Okay, so we will take, imagine that we are now able to measure 2 different traits in the common ancestor, trait X and trait Y. And let's imagine that these traits actually are not related at all. They just vary in a random way. So here I put a point at the original time, and then as time passes, you know, trait X will move to one side, trait Y will move to another side. This is what's called a Brownian motion, a random walk. You know, so you have this, and the more you let time pass, the more, you know, your point will be far from the initial point. Now we have these 2 species, so the common ancestor split into 2 different species. So now each of those species will have their own random walk. Trait X and trait Y will again vary completely randomly. And now we let again time pass, and we have this 3rd speciation event. So where each of these common ancestors will split, leading place, again, through random walks to new different species. Now, you have to see that if you have very little time, which as is the case of the latest branches in this tree, the phenotypes, although they vary randomly, they will not be very close from those of the ancestor. Okay, so now if you measure trait X and trait Y, you may have the impression that they correlate. So if you don't take into account phylogenetic information, you may be led to confound what's purely random variance, random change with some sort of meaningful biological correlation. So it's very important to take phylogenesis into account. This is, again, an example of what would be a Brownian motion. Here we have, again, the phylogenetic tree, simulated phylogenetic tree, and then the change in one phenotype at the beginning. So they all change randomly. At some points, they split. Then the lines change in color. And you see that the more we wait, the more the 2 different species will be different. Now, there's, of course, some other models that take selection into account. This is what's called the Ornstein-Uhlenbeck model of phenotypic evolution. And here, in this case, there is a selective value. We could imagine, for example, that brain volume, and many people tend to suggest that brain volume provides a selective advantage. So if you have a trait that does provide a selective advantage, you will observe this sort of convergence. You know? Now, there seems to be a value, so the larger brain volumes in this case, or this is just a random phenotype, but they would tend to be selected, so the phenotypes will be attracted by this better value. So we tried to look at what would be the case for primate brains, and we had the chance of being able to collaborate with people from the Natural History Museum here in Paris, and also from the Institute of the Brain and the Spinal Cord, where we did MRI scans of a lot of different species. This is some of the species. Some are like more than 1 century old, and etc. And we managed to collect samples from 34 different primate species, which are represented by 65 different individual brains. And then this is the work of Katja Hoya and many more collaborators. These are some of the reconstructions that we were able to create from the MRI data. They are all at the same scale. So you have, for example, the human brains, all the great apes, chimpanzees, bonobo, gorilla, orangutan are on the bottom row. And then as you go lower down the figure, you get to see the smaller brains, and then you get to see also this striking relationship between brain size and folding pattern. What's also very interesting is that if you look at the human brain, the folds on the left hemisphere and the right hemisphere, although there is some resemblance, there tend to be a lot of asymmetry. So the human brain and large brains are known to be asymmetric. On the contrary, if you move to the smaller brains, they tend to be much more symmetric. So we developed, in particular, some methods for being able to estimate the what we call wavelength, which would be the thickness of a fold. No? Here it's what's represented on figure C to the right of your screen as W. So and here you have the formulas, and we even have the code that would allow you to estimate wavelength. Now we use this phylogenetic tree that we obtained from genetic data. This shows the relationship, the phylogenetic relationship between all the 34 different species, with some of them represented by the little brains. And here you get to see, in particular, so all the branch of the great apes with orangutans, humans, bonobos, and then on top you get to see the lemuridae, and then the axis on the bottom of your screen represents time. So the common ancestor would be somewhere 75 million years ago. And then, for example, the split between humans and chimpanzees would be somewhere around 9 million years ago. So we can use now the statistical models like the Brownian motion model. We tried also the Ornstein-Uhlenbeck model, the early burst model, to try to see which one would fit better the data of differences in brain folding, brain volume, surface expansion, and also the wavelength of brain fold. So here you have, for example, the reconstructions through the phylogenetic trees of cerebral volume. And again, you see in red all the great apes that have a very large brain volume. And then you get to see also the lemuridae that have, so in dark blue, the smaller brain volumes. And now you can follow through the deep into the phylogenetic tree, and that will give you an approximate estimation of what would be the cerebral volume of the different common ancestors, and also the absolute gyrification index to the right of your screen. And it's very interesting to observe, for example, the case of humans. I don't know, I will quickly jump. So in the case of humans, you see that we go from something that's not the deepest blue. That's also very interesting. So if you look to the left of your screen, the prediction for the common ancestor is a brain volume that's larger than the brain volume of some living primates today. For example, the Galago-Demidov, on the first species that you have on the top of the phylogenetic tree, should have a brain that's smaller than the brain of our common ancestor 70 million years ago. So in the branch that leads to humans, from the common ancestor up to now, you have a constant increase in brain volume, but there are some other species where it's more varied. So you have changes that go up and down and up and down. That's, for example, the case of you will not be able to find it because I'm going too quickly, but that would be the case of the tufted capuchin, for example. Now you see that the cerebral volume and the gyrification index have a very similar pattern of change. It's because cerebral volume is very strongly correlated with the amount of gyrification. But to our surprise, that was not the case, not completely to our surprise, but that was not the case for the folding wavelength, the size of a brain fold. And this is, I think, quite an interesting finding. So we observed that for most species that do develop brain folding, because our method only works when there is profuse brain folding, but that would be like 2/3 of our species did present quite some degree of brain folding. The size of a brain fold is all the time about 1 centimeter. It doesn't matter if you have a brain that's 20 times smaller, the size of your brain folds will be about 1 centimeter. It's like some sort of a module. And I think that this idea is actually very interesting. So because of the mechanical, if we try to understand this within the framework of the mechanical morphogenesis hypothesis, and then we conceive the wavelength as related to the bending energy required to make a fold, then the cortical thickness of the cortex will be the main determinant of brain folding, and cortical thickness is well known to be pretty stable. So even in a mouse that's orders of magnitude smaller than a human, the average thickness of the neocortex is 1 millimeter, and in humans it's only 2.5 times larger, okay, for a brain that's massively smaller. So if cortical thickness is conserved, that will lead to a conserved cortical gyrification wavelength, which will lead to some sort of modularity, which will be all the time of the same nature, the size of a brain fold. So if we take all those parameters and try to estimate which of today's extant primates looks more like the common ancestor, it happens to be the II. So the II, you have the picture on the right. It's this very cute little animal, and that's what its brain would look like. So 70 million years ago, probably the brain of the common ancestor was something like that, with some folding and like not too small. It's a size that's probably similar to that of a ferret today. Now again, this folding pattern. Now, how do we get to understand this folding pattern? Is this folding pattern determined by some genetic? Is it selected? Could we detect traces of selection in the formation of one pattern of brain folding compared with another? And we try to figure out a way of quantitatively estimating the similarity between different folding patterns. This is the method that we came up with. So to the left of your screen, you see one surface reconstruction, that's a brain surface. And then what we do is we compute the curvature and we remove everything that's a sulcus. So the figure in the middle is like a Gruyere, like a cheese where all the holes are the places where a sulcus should be. Now we have another algorithm that's able to shrink all the gyri, so it transforms all the cheese somehow into a wireframe. And that's the image that you see on the right. That's like a wireframe of a brain where each wire represents a gyrus and the holes are the soul side. Now, this wire, what's interesting is that you can just take the fact that a link exists and represent that as a link in a graph. So we can simplify these wires into a graph. This is, for example, the graph for the tufted capuchin. So we take this wireframe, we flatten it. Each point is a node in the graph, and then each link is just the fact that you had a gyrus between those two positions. These are now we call this the fold graphs. These are the fold graphs of 9 of our different primates, and you can see some sort of a pattern emerging. We go from the galago that has very little folding to a gibbon that's a much more folded brain. So for reminding you, this is, if we take only the species for whom we computed the fold graphs, this is their phylogenetic tree. So with a divergence 75 million years ago, we have a branch with the galago and the II, and then another 2 main branches, one with the macaques and the rhesus macaque and the gibbon, and then in the middle, the capuchin, the ruculi, and woolly monkey. Now, and this is actually quite interesting. If we only compare, so when we have these graphs, there's methods for comparing how similar two graphs are. And the method that we use is called the graph edit distance. So the graph edit distance is how many changes do you need to do in one graph to transform it into another graph. So it will give you some sort of algorithm. For example, add a node here, remove an edge here, and then the amount of changes that you need to do, that's the graph edit distance, okay? So when you compare, if two graphs were exactly the same, then their distance would be 0. There's no need to make any change. But if you have something that has very little folding and that you want to transform into another fold graph with a lot of brain folding, you will need to do a lot of edits, and that gives you different graph edit distances. So if we compare the distance between one brain to the other, and that's the matrix that you have in the middle, now the more similar animals will have a smallest point, so a small distance. And you see, for example, and this I will show in the next, I will highlight just these two species, which I find particularly interesting. So you have the tufted capuchin to the left and the crab-eating macaque to the right. They appear, when you compute the similarity between the folding patterns, to be extremely similar. So in the graph in the middle, they appear right one close to the other. However, if you look in terms of phylogenetics, they are very different. So the branch that leads to the capuchin monkeys split from the branch that led to the crab-eating macaque probably some 50 million years ago. What's more interesting is that if you look at the common ancestor between the capuchin, duruculi, and woolly monkey, the brain of their common ancestor was very likely not folded or little folded, which, you know, here what you observe is that two brains, only because of their size, develop a folding pattern that's very similar despite their evolutionary history. How would it be possible if genes were the main reason for the encoding of a folding pattern, that a folding pattern disappears and then reappears to be exactly the same as in a completely different branch? Okay, so this for us, it's a signal that it's probably more likely the mechanical constraint, the mechanical morphogenetic effects that influence the formation of a folding pattern relative to the geometry and to the amount of growth than a very precise prescription of the folding pattern through a genetic program. So now for the conclusion. Within the context of an adaptationist program, natural selection would be the main optimizing agent during evolution. So it would be selection that would lead to the formation of one trait or the other. Organisms within this framework are seen as a mosaic of unitary traits, each subject to their own adaptive story. There's been an opposite kind of point of view, or alternative or complementary point of view, which is the idea of the BYOD plan, which suggests, by contrast, that an evolutionary process, every evolutionary process is constrained by phyletic heritage and developmental constraints. Okay, so there's somehow this idea of a plan that you can deform, but you can't really dramatically change one part without changing another one. After the modern evolutionary synthesis, which combine monogenetic and phylogenetic perspectives and polygenetic perspectives on evolution, there was an extended evolutionary synthesis which aims at further including processes such as developmental constraints and non-genetic inheritance and self-organization. Mechanical morphogenesis is an example of this sort of non-generic inheritance. You don't inherit a specific gene. What you inherit is the set of properties which are common to all mechanical tissue, and emergence is a clear example of self-organization. In particular, the concept of evolvability of an organism becomes an important property. Structures should be able to change and to adapt, but also sufficiently stable to conserve their fitness. For example, they should be stable vis-a-vis pathological variation. So if everything were encoded by genes, and if it's only one or a few genes that encode a trait, then a mutation, they would be very fragile to one or a few mutations, and it would be very difficult that an advantageous mutation, given a specific population, would be able to spread through the population because of the unlikely of the mutational events. However, there's one way in which evolvability can be greatly improved, which is through compartmentalization. The creation of relatively independent modules. You can very easily, the idea of a compartment and why they are important for evolvability is that you should be able to produce very easily a new module. Then each of these modules should share many of their properties, which would make them especially beneficial for some adaptation on top of that. And it seems, in our opinion, that mechanical morphogenesis fulfills these requirements. You are able to produce very robust modules, the brain folds, which are large scale. They can be regulated by a strongly polygenic factor, which is just basically brain growth, and that leads to a brain growth. So it would be very easily, by modulating this polygenic factor, brain growth, to produce the creation of an additional brain fold that would have the same mechanical properties, the same mechanical gradients as some other folds, which would make it easy for integrating them into selection and adaptation. So with that, I will finish my presentation. These are some of the collaborators whose work I presented, and the main idea, I think, if there's one to take, is that the idea of the spandrels that Stephen Jay Gould proposed and Richard Lewontin many decades ago. So if you try to build a cathedral on top of arches. Just by necessity, you have these triangular parts, which are where the arches support a dome. In the Cathedral of San Marco, where you see a picture here, they were used in such an effective way that you could get the impression that they were actually designed to be there. You could get the impression that the architects were asked to produce these spandrels, these triangular regions that play such an important role in the iconography of San Marco. But actually, we know that they are just a developmental constraint. So if you want to make a dome on top of arches, they will have to be there. And it's only after that the artists find a way of using that up. So if you look at the history of human evolution, modern humans have been around for probably 130,000 years, but the first indices of properly modern human behavior, like art and the creation of sophisticated tools, it is much more recent, probably 50 or 60,000 years ago, which means that we had, for maybe half of our evolutionary history, a brain like the one we have today, but which was not, it seems, able to perform the same functions that we're able to perform today. So it would be possible that structure actually precedes function before, instead of being the opposite. So thank you very much. And with that, I will finish sharing my screen. And you can, of course, get in touch with me. The best way to do it right now is through Twitter, but emails are also seem to work. Thank you very much.
Okay. Thank you very much, Roberto. It was a great talk. And well, there are some questions also. Alateya says, thank you so much for sharing your amazing work. Do you have any comments or recommendation regarding childhood, adolescent, adult development in humans?
Yep, we have actually been interested in autism for quite some time. And autism, there's many studies that try to relate that to problems in connectivity, which we could say it's the case for any psychiatric disorder. They are today conceived as a problem of connectivity. Many of the tools that we're able to use to assess the phenotypes in actual human childs or adults, are based. MRI is a fundamental tool. The problem is that most of our biological knowledge comes from mice. In particular, in mice, there's first of all no brain folding, and then the brain is so small, it's actually much smaller than what people have estimated to be the size of the common ancestor of all mammals. That there's not a very profuse cortical connectivity. So it's very difficult to try to, if the processes that we are trying to model here, so the formation of brain folding, a rich cytoarchitectonic organization, the formation of cortical connectivity, probably the only way of addressing that will be through gyroencephalic species and probably through magnetic resonance imaging. At the moment, we are mostly doing image analysis, and as I showed you, you can have the impression that you are observing one phenomenon, but if you don't have a clear theoretical or biologically grounded model of what's going on, you may be, confused by an artifact somehow. So very often, for example, this through pure image analysis, what I was showing you is that you would be able to observe a differential expansion of gyri compared with sulci, but that's not necessarily a reflection of a specific phenomenon going on there. That's very typical of dynamical systems, where it's the whole system that may produce something that looks like a localized effect without being such. So at the moment, our work, the combination that we try to do between this very theoretical basic science part and our more closer to the translational work, is try to approach the models that we use to what's relevant for the clinics. I hope that that's how I addressed the question.
Great. Well, you have a lot of congratulations.
Yes, that's very, very kind. I would like to, like, we have been investing quite a lot of time in making our tools available on the web. It's kind of what I presented also last time you invited me. I would be really delighted to work with all of you guys. Like, We try as much as possible to put all our data on the web. All our ferret data is on the web. All our primate data is on the web. The tools that you need, you don't even need to install them. They are just on the web browser. We are currently trying to increase the number of primate species from the 34 that I showed you to 60. And like, if you are interested in any of these subjects, like, please do get in touch with us. We will be more than happy to start collaborating with you. Like right now, from me sitting in Paris, the distance from my nose to my computer screen is the same as from your computer screen to your nose. I'm as close to you than to my neighbors here. Like, we're all confined. It's like it's an ideal opportunity, I think, for kickstarting transcontinental collaborations.
Thank you very much, Roberto. This is very generous. I mean, all the students in the workshop were delighted with your generosity, and now in the symposium also. Thank you very much.
Thank you.
But we have a couple, Angel Caputi, you know you're there. We have, I am a little bit delayed, but we had to change as I send you the link, the conference, because of some intruders. And so I'm going to have just two questions, last questions. There's significant brain, is Robert Monfera, fascinating, thanks Roberto. There's significant brain volume variability among individuals of a species. For example, humans, should it lead to different gyration or surface to volume ratios?
Exactly. And we actually looked at that in a paper in some years ago. Humans are quite interesting. So if you look at chimpanzees or macaques, or the difference between the smallest brain that you can observe and the largest brain is not very large. But when you look at humans, you can go in any like sizable sample size. You can have people that have half the brain volume of some others, all while being perfectly normal. It's not microcephaly and macrocephaly. It's just normal people, all like completely equivalent. But then you have these very dramatic differences in brain volume. And if brain volume is the main trigger for brain folding, we should expect that people with a larger brain would have more brain folding, which is actually what we observed. So if we take the smallest brain in a human sample, and we were looking at more than 300 different human brains, and you just scale it up so that it will have the same brain volume as a large human brain, then the cortical surface area would be 20% less than in a normally large brain volume. You will have less brain folding and less cortical surface expansion, actually. We also looked at the gradient of this folding. So it's very well known in when you look at the brain folding in primates, that primates tend to have a cortex that's more folded toward the back of the head compared with the frontal lobe. And for example, the main differences that you can observe between chimpanzees and humans in terms of brain folding would be in the frontal lobe. Although there's this, on average, all brains tend to be more folded in the back than in the front. And that's exactly what we observed when we compared only humans. So all members of the same species, normally with the same genetic makeup, kind of. So people with large brains tend to have, on average, a more folded brain. And this brain folding was particularly striking in the frontal regions. So what you are able to observe across species, you can also observe across members of the same species.
And well, the last one is, thank you for this nice talk. To question, what would be the mechanical role of the school? And also whether if tracing the shirification along development would allow to predict different growth rates on different regions of the brain. This is from Caputi, Angel Caputi.
Thank you very much. Yeah, that's a very interesting question. So very often when you read a neuroanatomy book, the main idea that people will be exposed to is that brain folding develops as a necessity to be able to fit the neocortex within the limited space of the skull. Okay, that's probably what most of us read when we opened the Candle book, no, or something like that. It's like the first basic idea, and it's false, and it's been known for quite some time that actually the skull doesn't play any role in the formation of brain folding at the beginning. So there's several examples. One that's quite nice is, well, quite nice scientifically, it's a baron in the 50s. He did a series of experiments with sheep brains. So he was able to remove, through aspiration, half of the brain of a sheep fetus before the onset of brain folding, and then let development continue. And what we observed, so now that you have this brain inside this little skull, and there's enough space for the remaining brain to grow without folding. There's no space missing or anything. There's a lot of extra space. However, the brain did fold, and the folding pattern was actually very similar to the folding pattern of a normal sheep. Okay, so since then, the idea was pushed forward that actually neocortical folding is intrinsic to the neocortex. Okay, then there's even some, you could think that it's because, you know, the aspiration is quite a dramatic intervention. It's very, very extremely invasive. But there's even in humans, and there's, I know of 2 papers that have made reports of people who are born with only one hemisphere. There's one paper in 2002, and another one that I wouldn't remember the date, but more recent. So this was the case of a girl that was born with only one brain hemisphere. So the hemisphere that was there could have developed without folding. There was plenty of space. It did not. It stayed in the half of the brain where you would expect a normal brain hemisphere to be, and it had a normal folding pattern. There were many functional weirdness. For example, all the visual cortex, instead of being separate into the left and right hemisphere, by necessity, everything was in just one single hemisphere, which was quite nice. But more striking, the girl was like she was the first in her class. No major cognitive impact of missing actually half of a brain. So, however, there seems to be a role. So many people, for example, in the study of evolution, the skull is very important. It's most of the time the only thing that we have. So at the beginning, when a brain is growing, it's actually the brain that pushes the skull. The skull at the beginning is very, very soft. In the case of humans, there's the fontanelle. So the bones in the skull are open. So they are soft, they are open. So the skull doesn't produce any major mechanical constraint. So it's the brain that pushes the skull, leading to its shape. In the case of hydrocephaly, for example, when the brain grows too much, the skull is just larger. But that will leave a trace of the folding pattern, that will imprint the folding pattern in the skull. And that's what allows us to try to get an idea of what the folding pattern of our ancestors looked like. So it seems that because of this folding that's already there, that's imprinting the skull, that will somehow, for example, very often the higher parts of gyri are not round, they are completely flat. Well, they are quite flat, and that may be because they were pushing against the skull. But that's a very late stage, not the onset of brain folding. So that's also the case, for example, for vasculature. No? Very often people observe that some main veins seem to follow the valleys in the sulci. No? They grow along the sulci. But it seems again that it's the brain folding that somehow influences the formation, the path that different veins will follow, more than being the other way around. And,
okay.
yeah. And then for the growth rates, yeah, absolutely. You can still observe brain growth until the age of 10 in humans, and it's very often a very regular asymptotic shape. And we are able to measure like the last traces of the differential expansion of different regions in the cortex in using magnetic resonance imaging in particular. There's a very interesting project right now. The data should become open at some point. It's called the Developmental Human Connectome, and they have at the moment data for the late stage of development, but they should be making available data in utero data for human brain development in MRI.
Well, thank you very much, Roberto. It was a great talk and also very interesting questions and also all the information you're sharing.